this is Christian Montalvo. This video is for about 225. This is video from the week number 5. It's called Biochemical Testing for Microbial Identification. Today we're going to be doing the carbohydrate fermentation testing. So this is part number 1, which is the incubation preparation, this is the whole video for this week's lab exercise. So if you guys can get close to here. Okay, so here we already have the vials, the broth, the red broth that we're gonna be cultivating our cultures today. And this is the way we're gonna do the fermentation test. Due to the time, I already mixed 2 of them, but I'm gonna mix the last one, which is the lactose. Which is the glucose. So we're gonna put half of this powder into each of these containers. One is for this bacteria, which is the cerevisiae bacteria, and the second vial is labeled as well, and this is for the epidermidis culture. So we're gonna put half and half of the glucose. So for us to do that, we're gonna I'm gonna put more light in here. So we're gonna open this and we're gonna sterilize it with the flame. Once it's sterilized, we're gonna open the glucose and we're gonna add half of it. Oops, okay. Half of it, and then we're gonna have to sterilize it again before closing it. And then we're gonna carefully mix it until it dissolves. So we see there is no glucose there no more. It's already mixed with the red broth that we have this week. Now we're gonna do the same thing with number 2, or glucose. All right. Then we're gonna add half of it. The whole, whatever is left in there. And then we're gonna sterilize it again. Okay. Now we're gonna close it. Okay. Now we're gonna mix it. Like I said, the other 2 I already mixed due to the time, but I left the glucose one so I can show it how I did it. See, it's already well mixed. Now, if we follow the steps, we're gonna insert one of the Durham tube into each phenol broth vial so that the opening of the Durham tube points toward the bottom of the broth. So we're gonna do that into the ones that we have here left. We already put it, sterilized it in alcohol, and we already let it dry. We already let it dry to air. So I'm just making sure. I'm gonna put this candle on the side. Just making sure it's all well dry. Okay, so they are dry. So now what we have to do, we have to open one of them and make sure the opening is towards the bottom of it. We just gonna slide it down. We have to make sure there's no air in the Durham tube. So we just go like that. Okay. And then we'll do the same thing for the other one. Just make sure there's no air. Well, there is air there. Okay. There you go. Okay. And now we're gonna add it to the last two that we have here. A lot of air. This one. And now we're doing the last one. This one's a perfect shot. Just trying to make sure there's no air. There's a big bubble of air there. Okay. Now that we have all ready to introduce the culture, what we're gonna do, we have a pipette. We're gonna pipette the culture with this pipette that we have here. So sterilize. And we're gonna introduce 4 drops of the active culture to each and one of the corresponding bacteria vials. So for them, we're gonna have to, this is what I'm gonna do. I'm gonna make sure that all of them in order with the right label. There you go. And what I'm gonna do, I'm gonna grab the right one too. What I'm gonna do, first I'm gonna sterilize each of these 3. I'm gonna put it back into the rack so I don't drop any. Okay, I'm gonna put it there. Try to do as quick as possible. Right there. I'm gonna grab the last one. Okay. That was the last one. Now I'm gonna open this one very quick. I'm gonna put it on this side. I'm just gonna sterilize it. Alright. Now I'm gonna pipette. Okay. I'm gonna close it again. I'm going to introduce 4, if you guys can get close, 4 drops of each one. 1, 2, 3. Need one more. 4. 1, 2, 3, 4. 1, 2, 3, 4. Okay. Now I'm gonna have to sterilize it again. One. 2. And the last one. 3. All right, and the same thing I'm gonna do with the other 3, which is introducing the epidermidis light culture into the red broth. And after that, we're gonna place the 6 inoculated phenol red broths into an incubation location, and then we're gonna leave it there for 12 hours. And then after 12 hours, you can leave it there between 12 and 24 hours, but no more than 24, because if it's more than 24, it may give us like a false result. So we're gonna do between 12 and 24 hours. And then I'm gonna take a picture and I'm gonna upload those pictures into the lab report. Okay, so thank you very much, and I will see you next time.